Thank you so much, Dr. Rahman. We'll move forward with our next talk to Dr. Alessandro Prigione, who will be presenting on pluripotent stem cells and brain organoids for Leigh syndrome. And please put your questions in the chat and we'll get to them at the end of the session.
Yes. Have a good afternoon, morning also from my side. Thank you very much for the kind invitation. So so exactly. So in our research, we are also focusing on Leigh syndrome, and we use pluripotent stem cell and brain organoids, which you see an image here in this picture. So thankfully, I don't need to go after wonderful Shamina's summary. I don't need to go to the details of the disease. I just want to point out that in our lab, we're at the moment mainly focusing on gene ATP6 and SURF1 because of the frequency that has been observed in patients. A bit also with respect to the severity, with SURF1 is quite severe, but also because we believe these two also have more urgency in terms of a modeling system, because we know for ATP6 is difficult, has been difficult to generate models, but we will hear about this maybe after my talk. And for SURF1, there are models available, but also has been challenging, even though it's a nuclear gene. So we think that this could be why there could be a need for a different approach. So, so what we do basically is what a lot of people do when they work with iPS cells, is that you start with your patient-derived material, and then you reprogram that. In our case, mostly is fibroblasts, so somatic cells from the skin. We reprogram to this induced pluripotent stem cell, which then can differentiate in every kind of germ cell, like an embryonic cell. And of course, since there's variability between lines, it's always important to make so-called isogenic lines. It means they're the same, but only where the genome is corrected. For nuclear genes like SURF1, we do that. It is possible. For mitochondrial gene is more complicated, and we will hear after my talk about what has been improved in that area. And what we do afterwards to generate this model system, in particular of the nervous system. So we make neurons and these brain organoids, which are three-dimensional structure. We resemble some features of the developing brain. I meant some because it's still a model, it's still not full brain, and there's a lot of input from different labs to increase the resemblance to an actual brain, and there are still something that is missing in terms of the vasculature and other cell like microglia or other components. So also myelination comes very late, so it's still something that it's been need improvement. And then with this model, what we're trying to do is to study the disease and also see if we can find any phenotype that we can use for screening to identify treatments, with the idea that we could then maybe suggest that to patient. Of course, that is always the most difficult part, which is so-called valley of death. For people working in basic science, we are maybe getting excited from basic discovery, and then it's very difficult to really translate it into effective in the clinical setting. So, and another thing that I want to mention, of course, is there are specific challenges for mitochondrial genome. Not only the mitochondrial genome editing, but also when we make iPS cells, so induced pluripotent stem cells, because if we start from heteroplasmic mutation, so that means when a certain mutation is present, not in all mitochondria, you see here, so the pink one are the mutated and the green are the non-mutated, you may have iPS cells that have different level, even if they come from the same individual. So in some cases, this is good. That means you might have lines without the mutation, can be used as a control. But in some cases, it means we cannot entirely recapitulate the patient mutation level. Of course, when we have cases where the original somatic fibroblasts contain only mutated mtDNA, so like homoplasmic mtDNA mutations, this is not the case. We obtain only iPS cell line with the full mutation. And so that means we can recapitulate the patient, but of course, there's also means we cannot have isogenic controls so easily unless we try to use this new technology like mtDNA base editing, which is still a challenge in iPS cells, has not been validated yet, so demonstrated yet. Yeah, so what do we do with these organoids? So when we look at SURFAN mutations, we saw that compared to control organoids, the SURFAN mutant ones show some defects in the size. And so here are some immunostaining. You see that in red, it is a marker for neurons. You see in the control how much red. So there's a lot of neurons developing, and in the patient, this is really defective. But not only this, you see also in green, this is a marker for progenitors. So are the cells again give rise to neurons and glia, and they are don't show the nice organization like the shown in the control. You see also here again in green these progenitors. You see in the controls they make these neurogenic zones from which then the neurons develop, which is also what is in the actual brain. And in the patient, this is not occurring. So we think that there's a so we think that SURFAN mutations may disrupt early development. So and normally Leigh syndrome has been always considered a neurodegenerative disease where everything is working perfectly and then start to degenerate. But we think there are something using this model, this suggests there might be something else occurring before the actual degeneration, suggested that system might be prone to this sort of decompensation. And we also now in unpublished work, we also look at ATP6 organoid, and we also see here in the control these nice neurogenic zones of this progenitor organization, which is very much disturbed in different organoids containing different ATP6 mutations. So also there, we think these neurodevelopmental aspects are then impaired by ATP6 defects. We also looked more particular at some area of the brain, because we know, we heard from Shamima that Leigh syndrome has also specific disruption in some area of the brain. So the basal ganglia, like containing like the midbrain, or the striatum, so containing specific neuronal population. And so we look at these from SURFAN, and we see not only this developmental aspect, but also some defect in how neurons are and the length of the neurons. So these are axons, so how the neurons communicate, and we think that that is disrupted. And so if that is disrupted, might also impair the communication between brain area. And so what we're doing here is that we're trying to make so-called assembloids to assemble different organoids of different region of the brain. And so here, for example, this is from the striatum, which is one region affected, commonly affected by these mutations. And we try to assemble it to cortical and then see the connection. And we're doing that, for example, by infecting with specific marker cortical organoids before assembly, and then looking how these things can be connected. And so then you can see that some of the neurons then can reach to the striatum. And we think that this might be affected in with these Leigh syndrome mutations. So we can then go, after learning this part, we can then go in two-dimensional culture and study this in more detail. So in fact, we have found that SURFAN mutation these defects can be reverted by overexpressing wild type form of SURFAN, which we've heard about gene therapy. So if you would overexpress wild type SURFAN, we see that there's a recovery in neurons of the basal mitochondrial respiration, oxygen consumption rate, but also of this neurite length. Also what I meant is length of the neurons. And we also confirmed that using AAVs in neurons with a mutant SURFAN, that again, after this AAV delivery of in this two-dimensional neuronal culture, there's a recovery of oxygen consumption rate and neurite length. So this suggests that what we heard about before from Stephen about gene therapy, we can do some study in 2D in vitro to determine efficacy. So basically, to summarize, we think that when we make from pluripotent stem cell neurons, we think normally, physiologically, there's a shift from a certain kind of energy, which is glycolysis, to mitochondrial respiration. And so we think that this can happen early, already at the level of progenitor cells. So they need to be able to commit to mitochondrial respiration in order to differentiate into neurons and then make the correct branching. And so in Leigh syndrome, if this is not happening and this failure they fail to commit to mitochondrial respiration, then neurons can be less, can be shorter or defecting the ability to make this branching. And and this can be then a target of intervention. And this is my last part where I mentioned some of the way how we can find therapy. So we try to develop some screening settings. So we have one based on neuronal branching. So we can use iPS cells, differentiate, and then basically measure how much after replating the neurons, how much they can regrow these branches, this length axon. And this we can do, we have some unpublished work where we do a screening in silico to identify first candidate drugs, and then we could go to our system to validate into neurons. And in this case is SURF1 mutant neurons. So we're identifying some drugs that can be repositioned. So here is an example that the mutant has less branching, and after the drug is an improvement. And the other screening assay that we have is for ATP6, where a few years ago, we found that neural cells with ATP6 mutation have a defect in the mitochondrial environment potential. So they are hyperpolarized. And so we set up a screening also for that. It's in live neuronal cells where we can measure mitochondrial environment potential with the live dye and then quantify. And based on that, we found some candidates that now we've done a first off-label compassionate treatment with one boy that then recovered surprisingly well after a crisis using this drug. And this was the first compassionate studies that we tried in 2018, and is still taking these drugs today. And we have tried and 4 other patients, all with different ATP6 mutations, all then under supervision of collaborator Marco Schulke at Charité in Berlin. So we'll not go into the details, but I just want to say that just very recently we got approval from EMA, European Medicine Agency, for this drug for orphan drug designation. So we are now working on publishing this paper as soon as possible. And we are already meeting with EMA for establishing a trial. We're trying to collect the funds. And yeah, so with that, I hope I give you a summary of what we're doing. We think that using this model, we can understand a bit of the mechanisms underlying the disease in how neurons or neuronal or the brain is affected. I told you about this neuroprogenitor organization. So the fact that the disease might have some neurodevelopmental features, and the fact that branching can be impaired and can be corrected in some way, for example, by gene therapy, and we're working on other drugs. And then I mentioned some drugs that I didn't mention the names because we're still unpublished. But I'm grateful for all the team collaborators and all of you for the attention, and for Cure Mito as well for funding and support. Thank you very much.